So you are a cell, and you want to replicate your DNA for mitosis. DNA replication is the process by which a cell copies its DNA so that each new cell receives an identical set of genetic information. This process occurs during the S phase of interphase, before cell division, and it's essential for growth, repair, and reproduction. Before we start, let's go over some basic rules of DNA structure. All DNA is comprised of 2 strands of DNA, which run anti-parallel to each other, and 4 nucleotide bases. These nucleotides pair together as such. Adenine pairs to thymine, and cytosine pairs to guanine. These pairs are held together by hydrogen bonds, which allow the DNA to hold fast while also being able to easily unzip. DNA replication is described as semi-conservative, meaning that each new DNA molecule contains one original parent strand and one newly synthesized strand. Let's begin. Replication begins at specific locations on the DNA called origins of replication. In prokaryotes, there is usually a single origin, while eukaryotic cells have many origins along each chromosome. Having multiple origins allows large eukaryotic genomes to be replicated more efficiently and quickly. At each origin, the enzyme helicase unwinds the DNA double helix by breaking the hydrogen bonds between complementary base pairs. This separation forms a structure called a replication fork, which moves along the DNA as replication continues. As DNA strands are pulled apart, single-strand binding proteins attach to the exposed strands to prevent them from re-annealing or forming secondary structures. Meanwhile, topoisomerase works ahead of the replication fork to relieve tension and prevent overwinding of the DNA. Once the DNA strands are separated, synthesis of new DNA can begin. However, DNA polymerase cannot start a new strand on its own. Instead, an enzyme called primase synthesizes a short segment of RNA called an RNA primer, which provides a starting point for DNA synthesis. The main enzyme responsible for adding new DNA nucleotides is DNA polymerase III. This enzyme adds nucleotides only in the 5' to 3' direction, using complementary base pairing rules. Adenine pairs with thymine, and cytosine pairs with guanine. Because the 2 DNA strands are anti-parallel, replication occurs differently on each strand. The leading strand is synthesized continuously in the same direction as the movement of the replication fork. In contrast, the lagging strand is synthesized discontinuously in short segments known as Ozaki fragments because it runs in the opposite direction. Each Ozaki fragment requires its own RNA primer. After the fragments are formed, DNA polymerase I removes the RNA primers and replaces them with DNA nucleotides. Then, the enzyme DNA ligase seals the gaps between fragments by forming phosphodiester bonds, creating a continuous strand of DNA. By the end of the process, replication produces 2 identical DNA molecules, each composed of one original strand and one newly synthesized strand. These DNA molecules are then ready to be distributed into daughter cells during cell division, ensuring that each cell receives the same genetic information. Before replication is complete, the cell must ensure that the new DNA is accurate. DNA polymerase III has a built-in proofreading ability. As it adds nucleotides, it checks each one for correct base pairing. If an incorrect nucleotide is added, the enzyme removes it and replaces it with the correct one. This proofreading function greatly reduces the error rate of DNA replication, making the process highly accurate, about 1 mistake per billion nucleotides. In eukaryotic cells, there is an additional challenge when replicating the ends of chromosomes. DNA polymerase cannot fully replicate the very end of the lagging strand. These ends are protected by repetitive non-coding sequences called telomeres. Each time the DNA replicates, telomeres become slightly shorter. To prevent the loss of important genes, some cells, such as stem cells and cancer cells, use an enzyme called telomerase to extend telomeres, allowing the cell to divide many times.